Right. So the most important part of this particular chapter is going to be aerobic respiration and its specific stages. When we actually get to the stages of aerobic respiration, the first thing that we have to actually discuss is the mitochondrion. Mitochondrion is the singular form of it. Mitochondria would be the plural form of it. Now, let me roughly draw a mitochondrion. So if I were to roughly draw a mitochondrion, it would look something like this. It's not a perfect oval, it's not a not perfect oval, it's almost something like this. Let me zoom in and I'm just going to roughly label everything I drew in. So here we have the outer membrane, which I'm going to call OM. And then we have the inner membrane inside. That would be right here. So in between, I'm missing the I. In between, we have something known as the intermembrane space. Nothing fancy. Two membranes. In between, we have an intermembranous space. Simple. What about this middle part? This is actually known as the mitochondrial matrix. So this is where all the circular DNA and solutions and other stuff is present. There are more parts to the mitochondrion, obviously, more detailed ones, but these are the very simple things that you have to be aware of. I'm also going to bring in this picture of the mitochondrion just for you to better compare. And as you can see here, we have our outer membrane, outer mitochondrial membrane, inner membrane here, and in between those spaces, we have the intermembranous space, and this is this matrix I was talking about. You see, everything is suspended in the matrix. That's the idea. Now, why exactly am I focusing on these parts of the mitochondrion? That is because we have to specifically talk about the stages and we have to actually be able to tell in which part of the mitochondrion these stages are actually occurring. So let's get to the schematic, the stages of aerobic respiration. Aerobic respiration is comprised of four main stages. Stage number one is known as glycolysis. You can call it glycolysis just to show that glucose is broken down, glycolysis, and it's just glycolysis is a better way of pronouncing it, I guess. No big deal there, right? Two. Stage number two is known as, I won't tell you. I'm gonna move on to stage three. Stage 3 is a cycle, and this is known as the Krebs cycle. Now, this is also sometimes known as the TCA cycle, that means tricarboxylic acid, and this is also known as the citric acid cycle. So, those are common names that we actually used back in the day. So, citric acid, I mean, not back in the day, we still use them. But according to your syllabus, you have to be able to refer to this as being the Krebs cycle, the Krebs cycle. So stage 1, glycolysis. Stage 3, the Krebs cycle. Why did I leave this blank? That's because this stage links glycolysis to the Krebs cycle. What did it do? It links glycolysis to the Krebs cycle. It links it. So this is simply known as the link reaction. That's why I didn't give it to you before. So glycolysis, then it moves on to the link reaction. The link reaction onwards to the Krebs cycle and then to the fourth and final stage. This final stage is where most ATP is produced and that is known as oxidative phosphorylation. Oxidative phosphorylation. Okay, it's known as oxidative phosphorylation. Like so. Oxidative phosphorylation. Now, to talk about the whereabouts of where these things are happening. If I take the cell, any cell has a cytoplasm, no? So actually this process of glycolysis doesn't really require the mitochondria. Now, I know that I told you that the stages are occurring in the mitochondrion, but actually this stage of glycolysis can just occur independent of the mitochondrion. It is just the breakdown of glucose. No, this is simply the breakdown of glucose into those molecules known as pyruvate, not to worry, we'll talk about that in detail in a bit. So that breakdown doesn't really require the mitochondria at all. That can simply happen in the, not the glycoplasm, the cytoplasm. It can simply happen in the cytoplasm. And then when it comes to the link reaction, Krebs cycle, and oxidative phosphorylation, we can't just say mitochondria, we have to specifically, precisely say where in the mitochondrion this is occurring. So for example, the link reaction actually occurs in the mitochondrial matrix. That inside liquid stroma type of thing actually occurs in the mitochondrial matrix. What about the Krebs cycle? Also the mitochondrial matrix. Same deal. The Krebs cycle will also occur in the mitochondrial matrix. Now you might think oxidative phosphorylation also occurs in the matrix, but not really. This is even more precise than happening in the matrix. Don't worry, when I get to the fourth and final stage, I'll elaborate on that. This actually occurs in the inner mitochondrial membrane. You can't just say mitochondrial membrane, we have to be specific. As we know, we have two membranes, outer membrane and inner membrane. So out of these two, this is actually occurring in the inner mitochondrial membrane. This is occurring in the inner mitochondrial membrane. Rough sketch, four reactions. First one's occurring in the cytoplasm, link reaction, Krebs cycle. Both of them are occurring in the mitochondrial matrix, this liquid suspension in the middle. And then onwards from the matrix, everything is moved to the inner mitochondrial membrane for oxidative phosphorylation to take place and the final ATP to be produced. With that, now we are more than ready to actually talk about all four of these stages in more detail. Let's start off with glycolysis. Glycolysis. Okay. Now, I'll start off with the 6-carbon molecule we call glucose. Call glucose. 1, 2, 3, 4, 5, 6. That is clearly 6 carbons. Henceforth, a circle will always represent a carbon atom. Please note that. Please keep that in your mind. So let's actually label this. That is glucose, and that is a 6-carbon molecule. Okay, what is the first thing that happens? The first thing that happens is that this glucose is attacked. By whom? Attacked by zombies, attacked by creepers, skeletons? Not really. This glucose is attacked. We call that attack as phosphorylation. So this glucose is first of all going to be phosphorylated by two ATP molecules. Phosphorylated by two ATP molecules. So what would that look like? Let me duplicate that. So that is our same exact glucose molecule. Now this is phosphorylated by two ATP. Like that. So I'm just going to represent the two ATP like that. That's phosphorylated by two ATP. Now this new molecule we have produced, we can't really call it glucose. It's no longer glucose. It contains extra ATP molecules. No, this new molecule that is produced is actually known as fructose 1,6 bisphosphate. Fructose 1,6 bisphosphate. That's what it's known as, fructose 1,6 bisphosphate. Now the thing about this is, after phosphorylation, this is not really that much of a stable molecule. It tends to break down. It tends to break down. So let's see what exactly this tends to break down into. So 6 broken down into 2 would be 3 each. No, boom, like so. This is broken down into 2. These things are known as triose phosphates. 2 triose phosphates. TP for short. You can just say broken down into 2 TP. Now remember, we had this ATP on that, we had this ATP on that. Now you have to actually know that this step has much more detail to it. This step is much more complex, actually, honestly speaking. Glycolysis, if you learn this at a master's or bachelor's level, so a higher study level, this is actually a 10th step reaction, or even you can consider it as being more than 10 steps. But in this stage at A levels, you only have to know the bare minimum of what glycolysis is. So the thing is, each of these steps are much more complex than this. This has been very simplified for the betterment of your understanding. Here simply there's a process that actually adds 2 more ATP molecules onto it. Otherwise you might get confused. So let's just clarify things. Phosphorylated by 2 ATP. These are those 2 ATP molecules on the triose phosphates as well. Okay, but do you see there are 2 more ATP molecules? There are 2 more ATP molecules. This stage actually adds 2 more ATP molecules, but we don't really discuss about that. We don't really make it that obvious. Why, you might ask? Don't ask, it's more complicated. But in this stage, all you have to know is that you have to consider that 2 extra ATP has actually been added onto it. So then the final step can occur onto these things. Now this is what happens, and this, oh my god, my line didn't straighten up. Right. So this is the final products of glycolysis. Now those are 2 triose phosphates. No. Now we finally get this completely broken down molecule. No ATPs, no phosphorylation, nothing. So we have our final 2 pyruvates. We have 2 pyruvate. So now for this to occur, both of these things have to be gone. So those things are actually removed. So we say that we produce 2 ATP. Now those things are released. Same thing here. We are releasing these things. Releasing 2 ATP like so. 2 ATP is removed like so. Very simple. Now at the same time, something else is occurring. To talk about this something else, I'm going to have to introduce you to a new concept, not really a new concept, but this is the story of coenzymes. Of course, we know about enzymes, a very common topic, but what about coenzymes? Simply put, very simply put, coenzymes are helper enzymes that can support metabolic reactions. For example, cellular respiration. Now, for example, you know, DNA replication, protein synthesis, those things cannot be done without enzymes, which you learned at AS level. You know, you need DNA helicase, DNA polymerase 3, Taq polymerase, RNA polymerase for all those reactions to occur. Now, coenzymes are also a set of enzymes that are going to help this particular reaction. When it comes to cellular respiration, you have 2 specific coenzymes you need to know: NAD and FAD. The full forms of these are actually nicotinamide adenine dinucleotide and flavin adenine dinucleotide. Why am I not giving those to you? Because those are not required by the syllabus. Actually, you know what? On second thoughts, nicotinamide dinucleotide. I missed the adenine, nicotinamide adenine dinucleotide, and then this is just the F stands for flavin adenine dinucleotide. Nucleotide, right? These are the full forms. I'm going to get rid of them. Dinucleotide. I'm sorry, my keyboard is so far away from me. That's why I'm struggling to type. Okay. Flavin adenine dinucleotide, nicotinamide adenine dinucleotide, FAD, NAD. I'll get rid of that. Now, the funny thing about these things are that they love to be reduced. They love to be reduced. Now, when it comes to your chemistry knowledge, you know that if we add hydrogen, we consider that as reduction. On the other end of the spectrum, if we add oxygen, we consider it as being oxidation. So if we add hydrogen, we consider it as being reduction. So actually they like to get reduced. They like to add hydrogen onto them. So FAD also. Now the funny thing about FAD is it can add two hydrogens. It can add two hydrogens onto itself. NADH can only add one, but this is not that important, so I'm not going to talk about that in much detail here. I'm not going to talk about how FADH can add two hydrogens, but do keep it in your mind at the basic metabolite level. Flavin adenine dinucleotide is capable of holding on to two hydrogens. Okay, when this happens, we typically refer to them as reduced NAD and reduced FAD. We typically refer to them as reduced NAD and reduced FAD. But why? Why do we need these things? Why are they so important? That will be made obvious at oxidative phosphorylation. For now, all you have to know here is that two of these coenzymes, so NAD, that's one NAD, that's another NAD. Two of these coenzymes actually interfere with the reaction, this reaction like this, and they become reduced. They obtain hydrogen from that reaction and they become reduced NADH. If this does not make sense at all, that is perfectly fine. Keep it as is. We'll talk more about that in oxidative phosphorylation. But for this step, all you have to know is we're losing 2 ATP from each of these molecules, and a NAD gets reduced at each end, just like that. So to just get an overall scope of the products what we have gotten, let's see, let me pick a nice color. Orange should do. Okay. So off the bat, per glucose molecule, we have obtained two pyruvate molecules. We have obtained two pyruvate molecules. And then we have obtained two reduced NAD molecules. If you get the idea, we have obtained two reduced NAD molecules. And the third thing we have to talk about is ATP. Now it's a little funny here. You see, now how many ATP have we actually produced? So here we get 2 ATP out of it. Here we also get 2 ATP out of this. So if we're considering this entire reaction, how many ATP have we produced? We have produced 4 ATP. But we don't really say that we have produced 4 ATP. Why? Do you remember that when we started this particular reaction, glucose was first attacked? Glucose had to first be attacked by ATP. So it was phosphorylated by 2 ATP molecules here. When that happened, we utilized 2 of them. So here now we produced 4 minus 2 would be equal to 2 ATP. So typically how we state this is that we say that we have a net gain of 2 ATP. We have not produced entirely 2 ATP. We have produced 4 ATP, but since we utilized 2 ATP from the start, we say that there is a net gain of 2 ATP. Be familiar with this. This step we will do for every single step. We'll be talking about what we obtain and how we obtain it. So for glycolysis, that's the complete reaction. This is a much more complex reaction, okay? Y'all are very lucky to be only talking about these couple few steps. Okay, pray to God, thank God that you don't really have to remember any of the enzymes, because believe me, the names of these enzymes are just out of this world. So just, yeah, all you have to remember is that this is glucose, this is fructose 1,6 bisphosphate, you've got triose phosphates, and then the final product, pyruvate. Now it should become obvious and abundant to you that when glucose is broken down by these specific steps, you get to pyruvate. You get to pyruvate molecules. Now, when you actually first encounter this word, it might be weird to pronounce it, see it, and write it, but you trust me, you'll get used to it, because pyruvate is going to show up a lot. I mean, a lot in your syllabus. Okay, right. Now, what's the second stage of aerobic respiration? The second stage is actually known as the link reaction. Now here's the funny thing, you cannot really move into the link reaction and you cannot move on to the Krebs cycle straight away. That's not possible. The only way that you can move on to the link reaction and then the Krebs cycle is if, a little bit of ICT here, if O2 equals true. What does that mean? That means that you can only move forth with the link reaction and the Krebs cycle if oxygen is available. Or in other terms, if this is actually aerobic, if conditions are aerobic. That's the purpose. Now we're doing aerobic respiration. If only oxygen is available, can we move forth? Otherwise, if we don't have oxygen, this glycolysis is going to just repeatedly occur, and just produce 2 ATP and 2 ATP and 2 ATP. That is not enough at all. In aerobic conditions, in aerobic respiration, we need oxygen. Only if oxygen is present will we move on to the link reaction and the Krebs cycle. So let's presume that yes, this condition is true. So we do have oxygen and this is a perfectly aerobic condition. In that case, we will be moving on to the link reaction. Now, do you realize how I have just selected one pyruvate? We produce two pyruvate, but you see, from this point onwards, we will only be using one of these pyruvates. Now, the link reaction and the Krebs cycle, it's occurring for both, okay? Both of these pyruvate molecules can enter the link reaction, the Krebs cycle, and oxidative phosphorylation to produce energy. For our simplicity, for our benefit, we will just be considering this. We will just be considering one pyruvate. So at the end, we're doing this. We will multiply everything by 2 at the end to talk about this pyruvate as well. Now, if that made no sense, completely fine. Throw that out of your head. Let's focus on the link reaction. Okay, I took one pyruvate all the way down, and the link reaction is very simple. Nothing special happens. Here, we're just trying to make this pyruvate able to enter the Krebs cycle. Now, why do we call it the Krebs cycle? Because it's a cycle. The Krebs cycle because it's a cycle. It's a recurring cycle. So this pyruvate cannot enter the Krebs cycle as is. That's not possible. We have to convert that pyruvate into a special something for it to be able to actually enter the Krebs cycle. So let's see what that something is. It's very simple. So that's pyruvate. Now the first thing that's going to happen is that pyruvate is going to be converted into a 2-carbon molecule. Now this is simply known as an acetyl group. Acetyl group or acetyl group. Simply known as an acetyl group. Now do you see 3 carbons to 2 carbons? What should have happened? A CO2 has to have gone. What did I tell you that this process was called? Now we'll write it down. This is actually known as decarboxylation. This is actually known as decarboxylation. Decarboxylation to produce an acetyl group. And in comes one of our friends. What do you think is coming here? Whenever we draw an arrow like this, it has to be an coenzyme. It has to be a coenzyme. Here we have our trusty NAD coming, interfering, and getting reduced to produce NADH, reduced NAD. Now we have an acetyl group. All good. Now, is this the thing that can enter the Krebs cycle? Can the acetyl group enter the Krebs cycle? The answer is still no. The acetyl group on its own still cannot enter the Krebs cycle. This acetyl group has to be joined with this guy. Now, do you see how he has a perfect opening for the acetyl group? So him, we call him CoA, not CoA. That's CoA. Elaborated, that would be coenzyme A. We have coenzyme A. So when the acetyl group joins with coenzyme A, we have this structure. So that's that, and that is the acetyl group. And this structure combined, we call it acetyl-CoA. Very simple. We're combining the acetyl group with coenzyme A. Now we have acetyl-CoA. This guy is perfectly capable of entering the Krebs cycle. No issues. He can enter the Krebs cycle. That's the only part of the link reaction you have to know. Now let's do our usual thing. Let's see what is produced. Now see here, the only thing that was produced was an NADH. Now, yes, we did produce a carbon dioxide. We will not talk about that. We'll talk about the coenzymes, if there's any pyruvate, and ATP only. So any ATP produced in this stage? Not really. So this is a good point to tell you. During glycolysis, ATP is produced. During glycolysis, ATP is produced. Now, in advanced level biology, there's not really MCQ. So you will not be writing paper 4 or paper 5 expecting MCQs at all. But when it comes to my revision segments, I do incorporate MCQs, specifically made MCQs for this chapter and every other chapter as well, because MCQs really help you solidify your knowledge on the concepts. It's almost a form of active recall and spaced repetition. Not to worry if you do enroll with the revision sessions as well. And if you follow through with these videos that I'm uploading for this specific course, you will have that revision within you. And it's a very interesting way that I have for you to actually revise with MCQs and flashcards, all happening in the future. And if you have any questions and if you want to talk about anything specific or you want resources, do hit me up. I am available in WhatsApp, Instagram, TikTok, Facebook, Pinterest, you name it, you can find my account entirely. It's by my same exact username that you find these videos under. I am always open to suggestions, comments, and anything. I will help you out in any form of way you require. No issues. Okay, getting back to this. So the only thing that is produced is NADH. So I'm just going to write down that. And NADH was produced. So one NADH was produced. Right. Now for this pyruvate scene. Here we just took one pyruvate. No. So for one pyruvate, we got one NADH. Now if we're considering the entire glucose molecule, if we are considering the entire glucose molecule, we have two pyruvates being produced. So when it comes to both of these pyruvates, we will get another NADH. So for two pyruvates, we will get another NADH. So we will say that we have two NADH. So overall, one NADH times 2. For the entire glucose molecule, we will have produced two NADH molecules in the link reaction stage. The link reaction state. So here under these almost like boxy things, we're talking about per glucose. We're talking about per glucose molecule. So here, obviously, per glucose molecule, we got 2 pyruvate, 2 NADH, and there's a net gain of 2 ATP. Here, just 1 NADH per pyruvate. Each glucose molecule produces 2 pyruvate molecules. Therefore, you simply have to multiply things by 2. And now we know that we have 2 NADH molecules. Hope that is clear. That's a very important distinction that you have to understand. It's just 1 NADH per this pyruvate, but actually per this glucose molecule, it's 2 overall NADH molecules. Right. I'm kind of going to throw a line like this just for clarity. Okay, cool. Okay, moving on to the Krebs cycle. The Krebs cycle. Like I said, this is also known as the TCA cycle, citric acid cycle, and this is a very major metabolic path. You have to know that. It's important to know. The Krebs cycle or the TCA cycle is such an important metabolic pathways because this is a pathway where so many things converge. What does that mean? So here's the central pathway. And here we are reducing, and metabolites are being produced. So many substrates from different sources and different parts of the body, different metabolites can actually come together to promote this process and be reduced, oxidized, and produced into energy. So this Krebs cycle is very important, right? So now we talked about how acetyl coenzyme A was the one who's capable of entering the Krebs cycle. So I'm bringing him here. I will bring him here. Okay. The Krebs cycle is confusing. It's a cycle. Cycles are confusing. And like I said, with glycolysis, you don't really have to know the entirety of the Krebs cycle. You don't have to talk about the Krebs cycle in too much detail, in an unwavering form of unwavering amount of detail. You don't have to do that. You have to know about just specific steps, and I will outline those specific steps in the exact same manner, in the exact same form that you have to know those from. So whatever compound I give at any given point at any location, you have to remember it as is. Don't try to mess with things and switch orders and turn it into an entire different cycle. That's going to take a toll on you and you will not be able to really understand the Krebs cycle. I'm going to give this in a very specific way, and that has been the best way I have found so far to help anyone understand. So I have tried this with so many of my students, and this was the best way that I have actually come across that's going to help you learn this. Right? So let's get to the cycle. Now we're starting off the Krebs cycle with something known as oxaloacetate. Now we're going to be introduced to some new compounds here, so you're going to have to bear with me and you're going to have to memorize those compounds. So we're starting with something known as oxaloacetate. What is that? Oxaloacetate. Oxaloacetate is actually a 4-carbon compound. Oxaloacetate is a 4-carbon compound, and that will join with our trusty. Thought I picked red. Come on, red. No, it's not really red. It's like a pink hue. Right. So this is going to join with our trusty acetyl coenzyme A. Where exactly is acetyl coenzyme A coming from? It's coming from the link reaction. Boom. And how many carbons does acetyl coenzyme A have? 2 carbons. How do we know that? Just double check. See? 2 carbons. And why in the world did this turn into an arrow? 4 carbons, 4 carbons, right. Acetyl coenzyme A. Now 4 plus 2, basic math. What is that? That would be 6. Now when these two things join, we are actually producing a 6-carbon compound, and no, don't think that we're producing glucose again, because we are not. Here we're combining these two things to produce something known as citrate. Something known as citrate. citrate, citrate, and this is actually why this is known as the citric acid cycle, because citrate is involved. Okay, first line of things, nothing else is here. That's the only set of things you have to know. There's this compound known as oxaloacetate that is combining with this acetyl coenzyme we're bringing in from the link reaction to produce something known as citrate or citric acid. Okay, now this is where things get interesting. This is a pretty straightforward thing, but now this is what is going to happen. Now here, 4, 2, 6. Now we're actually going to produce a 5 carbon compound, and this 5 carbon compound has an interesting name. This 5 carbon compound is actually known as alpha. My alpha went for alpha ketoglutarate. Now, full disclosure, your syllabus doesn't really want you to know this. You don't have to know that the name of this 5 carbon intermediate is alpha ketoglutarate. But without knowing it, this Krebs cycle is going to be more confusing. Hence why I have included this in all my teaching so far. So here we're going from 6 to 5 carbons. If you recall, we are losing a carbon. How are carbons typically lost? As carbon dioxide. So what is the process known as? This process is known as decarboxylation. So a decarboxylation process is occurring. And at the same time, one of our friends are interfering. So we have an NAD being converted into reduced NAD. So NAD being reduced into NADH. Here's a 5 carbon compound. That's the first place where we have all these coenzymes and other stuff being produced. The conversion of citrate, a 6 carbon compound, into the 5 carbon intermediate known as alpha keto or ketoglutarate. Right. And then here, obviously, from 5 to 4, we're going like this. That's the cycle complete. Do you see? That's a cycle. Now, I know I've drawn it like a rectangle. If I actually drew a cycle, it's going to be really hard to remember where the compound goes and what compounds come. It's going to be confusing. So I have done, this is also a cycle. No, I think you see? See? That's the type of cycle I typically have taught so far. Right. Now, obviously, 5 carbons to 4 carbons. What happens? We have lost a carbon. We have lost it as carbon dioxide. I'll write that down there in very small letters like so. Okay. When now a small tip I can give you is that whenever this decarboxylation process actually occurs, so whenever decarboxylation occurs, actually there's almost always going to be NAD involved as well. So here's our friend. So NAD is being reduced into NADH. NADH. Now the interesting thing here is that we are actually reducing 2 NAD to produce 2 NADH molecules. 2 NADH molecules. And we are also producing ATP. Now we are producing ATP. So here's the arrow. No? The arrow is heading this way. So I'm going to produce ATP like this. How are we producing ATP all of a sudden? I thought we had to wait until the final stage. No, I thought we had to wait until oxidative phosphorylation to actually produce ATP. I mean, yes, that is the idea. But now let me go back all the way to when we talked about how ATP can be synthesized. If you recall from spread 12.1 where we talked about energy, we actually talked about how ATP can be synthesized in two major ways. Number one was substrate level or substrate linked phosphorylation. I told you not to bother about it because when I explain the stages, I would get back to it. Well, here we are. And the second stage is chemiosmosis. This is in oxidative phosphorylation. Now focus on this. This is the primary way, I mean, not the primary way, this is the first way that ATP can actually be synthesized. This is known as substrate linked or substrate level phosphorylation. Do you see? Here simply we're linking some substrates. At this level, we're linking ADP, so adenosine diphosphate. We are linking it with a phosphate. So you see, adenosine diphosphate means two phosphates, no? So when we get another phosphate, it becomes three phosphates. So what is three phosphates? That's triphosphate. ADP plus inorganic phosphate produces ATP, like so. Okay, produces ATP like so. ADP plus inorganic phosphate produces ATP. Boom. That's the thing. And you might have thought that it's the final thing. Now pay attention to where I have located these as well. Do you see the NAD goes here? So always try to practice. The only way to practice these cycles and these processes is to constantly recall them, write these cycles, use a whiteboard, use a blackboard if you're still using blackboards. Use pen and paper, use a pencil. You just have to draw, write until your mind gets used to the idea of all these cycles. And whenever you're doing that, you try to do it at the same place. So you draw NAD, NADH here. You say that carbon dioxide is going away from. It doesn't have to strictly be like that. But the more your mind produces a repetitive thought, the more chances there is for it to actually stick in your mind. That's something regarding all these studying and everything. The moment that you can associate something in the exact same fashion in a million ways at a million times, the more you can do it, the better your chances at remembering it. That's the thing. So here you remember carbon dioxide is going off, and like I said, at any decarboxylation event, there's a very likely chance that NAD is actually involved. So here the thing is there's two NAD producing two reduced NAD, and this is the first scenario of substrate level phosphorylation. You see here we also produced ATP during glycolysis. We also did produce ATP. There was a net gain of two ATP molecules, but here we didn't combine any ADP molecules with inorganic phosphates. There was nothing like that. It was phosphorylated. That phosphorylated ATP was removed, and that's just this net gain. There was nothing happening in the link reaction, no ATP involvement, just NADH. But here we have the first ATP production by substrate linked or substrate level phosphorylation. That's the idea. And this cycle, the Krebs cycle, will also be the first scenario in which you encounter our second coenzyme. So this is actually where FAD gets reduced to FAD. It's actually FADH2, but I'm going to leave it as FADH like this. So it's FAD being reduced to FADH. FADH. Okay. A funny way you can remember this end. Now this end is easy to remember. An interesting way you can remember this end, something I still remember. I utilized this a long time ago when I actually did my A levels, but I still remember this. I used to remember this end as F tuna. You know tuna, right? Tuna tartar. Those weird things. I don't know if I'm pronouncing that right. Yeah. You know tuna, like the salmon thingy. I think I'm very bad at tuna, but never mind. So F tuna. There's one FADH, two NADHs. There's one FADH, there's two NADHs. Like F tuna, just to remember this. Two NADH, one FADH. That's all. Sadly, there's no way to remember the ATP. You can kind of remember it as this. There was one ATP, but it's about the coenzymes. Okay, like so. Let's do our typical final analysis. Let's see. Overall, we can see this 1 and 2. So that is a total of 3, wrong color, wrong color, wrong color, wrong color, 3 NADH molecules. Okay, 3 NADH molecules. And then we have produced one FADH molecule in the process, where right over here. That's the first occurrence of FADH, and we have also produced an ATP molecule. We have also produced an ATP molecule. Now remember, This is, so we have to do this box thingy per glucose, per glucose molecule. Here, all of these are produced per acetyl coenzyme A. So this acetyl coenzyme A, if we link back, is produced per pyruvate. But actually, in reality, if a glucose molecule is involved, there's two pyruvate molecules. Therefore, just like before, we gotta multiply all of this by 2. Per glucose molecule, we have to multiply everything by 2. So now we have actually produced a total of 6 NADH molecules, and we have actually produced a total of 2 FADH molecules, and there's a net production of 2 ATP per glucose molecule. Here we're writing the per glucose scenario. That is how many things, how many coenzymes, how many ATP is produced per glucose, like so. Now, just for a little overhaul of everything so far. If you see here, we have 6 NADH molecules. Here we had 2 NADH molecules, and there was also 2 NADH molecules produced here. So there's an overall amount of 10 NADH molecules we have produced, and there's an overall number of 2 FADH molecules that we produced per glucose molecule. So if you were to take the sum of all of these reactions, all these coenzymes and all these ATP molecules produced out of all three of these reactions, this is the final sum. 10 NADH, and there's a total of 2 FADH molecules along with 2 ATP basically. But we'll forget about ATP right here. We'll just focus on the coenzymes because those are the ones that are going to be important, and why and how, let's answer that now. So I'm just going to draw a line like this to separate things out, and now entering the final thing. Stage number 4, this is known as oxidative phosphorylation, and this is perhaps the most important part. Oxidative phosphorylation. Now, why am I saying that this is the most important part? That's because the most energy, most ATP is actually produced during this. There's no other importance of this. Okay, oxidative phosphorylation. Now, something you should notice that, see here. Here we took the pyruvate onto the link reaction. Here we're taking the acetyl coenzyme onto the Krebs cycle. There was always this chemical component that we took from one stage to the next stage, like so. But here, do you see? We're not taking anything. We're not taking the citrate. We're not taking the alpha ketoglutarate. We're taking nothing. This is an enclosed cycle. It can happen on its own. We're not taking anything. So then, what's the use? What's the use of all these reactions, and why? Here, these are the things that we need. The coenzymes. Huh, why? Let's see. So just to put it into your mind, we are taking 10 NADH molecules and 2 FADH molecules, which were produced from all these metabolic reactions. As we talked about before, this final stage is actually happening in the inner mitochondrial membrane, not the matrix, the inner mitochondrial membrane. So just for reference, okay, wait, hold on, let me write down that we're bringing in 10 NADH, 2 FADH total from here, right? This is my mitochondrion, and I'm just going to draw this, something like that. Okay, zoom in. I'm going to focus on this bit. Okay, I will focus on this bit of the inner mitochondrial membrane, and I will draw it in a larger fashion. And that will look something like this. So I'll use the grid for reference, and and I don't know, don't straighten it. Okay, there we go. That's it. That's just that part of the inner mitochondrial membrane. So this is the inner mitochondrial membrane. Of course, this is the outer membrane, this part, and this is the intermembranal space. I will get rid of those labels just to remove clutter like that. Okay, now another reminder. We are bringing 10 NADH and 2 FADH into here. 10 NADH, 2 FADH per glucose molecule onto here. Now, what exactly is happening? So, inside these mitochondrions, or inside these mitochondria, we have these inner membranes lined with these things. Inner membranes lined with these things. Now, these are electron transporters, and we typically call this the electron transport chain. Typically call that the electron transport chain. Now, actually speaking, this is the deal. These coenzymes, when they got reduced, or when they got reduced, they obtained electrons. Remember oil rig. So when they got reduced, they actually gained electrons because reduction is the gain of electrons. So now these NADHs and FADHs have plenty of electrons in them. So what do? They empty their electrons onto the electron transport chain. That's what happens. They simply empty those electrons onto the electron transport chain. So these electrons travel through the electron transport chain like so. The electron transport chain is built for electron transport. Very simple. So it goes along just like so. And something else also happened. Now these NADHs and FADHs have the H part as well, no? So they are removed as H pluses as well. They are removed as H pluses as well. And tell you what, so when these electrons actually go through the electron transport chain like so, like this, these proteins, these are also proteins, no? These electron transporter proteins gain energy. They obtain so much energy that they are capable of pumping these hydrogens, pumping these protons, those are H pluses, no? We typically call them protons. So capable of pumping these protons within the intermembranal space. Now, as a result of that, so many electrons are being emptied onto the electron transport chain. These NADHs, FADHs are losing the Hs, so that becomes H plus. There's so many protons, so much energy, and there's so much pumping going on. So many protons are pumped to the intermembrane space. So here now we have a lot of protons inside the intermembrane space, like so. There's a huge number of protons now inside. Why? Because of these electron transport chains actually pumping these things onwards inside the intermembranal space. Intermembranal space. That's the first set of things that you have to understand. It's the first set of things that you have to understand. Electron energy is being utilized by the electron transport chain proteins to pump H plus ions or protons from the mitochondrial matrix to the intermembrane space. Now, I will clear this first part. I'll tell you what exactly happens to these electrons and get it out of the way, and then move on with the rest of this reaction. Okay. Now, you know how when it comes to aerobic respiration, we say that we need oxygen. We say that we need oxygen so much, but why? So it's like this. Now, do you see this electron is not really being utilized by anything? This electron is not really being utilized by anything. It's just emptying its energy and it's just there. That's not good. We can't really have any free radicals or free electrons or any free charged particles. We can't really have them lying around. That's going to be very problematic. So what actually happens is that this is where this oxygen comes into play. Oxygen will act as the final electron acceptor. Oxygen will act as the final electron acceptor. Oxygen will act as the final electron acceptor. Okay, so this oxygen and electrons plus something else, I'll talk about this something else, can produce what? Actually, that something else is H plus, no need to gatekeep, but there's a specific way how this H plus actually comes here. We'll talk about that. I will gatekeep about that, but all you have to know is this oxygen combines with H plus and these electrons to produce water. If we go back all the way, where is it? Where is it? Where is it? Right here. We know that a product of aerobic respiration is actually water, right? We saw carbon dioxide being produced in different places, decarboxylation, that's how you get the carbon dioxide, and this is how you get the final water. Oxygen serves as the final electron acceptor, and that oxygen also combines with hydrogen. What happens when you combine oxygen with hydrogen? You get H2O, water. Okay, that's the schematic of what happens to these electrons like this. Now here there's a huge problem. When so many protons are pumped like this, we produce something known as an protonic imbalance or a protonic gradient. A protonic imbalance or a protonic gradient. See, what is this imbalance? There's so many H pluses inside, not that much outside. So it's an imbalance. We don't really typically like imbalances. No. Now the problem is they can't go across the electron transport chain proteins at all. And they cannot just cross the cellular, in a mitochondrial membrane to move into the matrix. That's not possible. There's only one way that they can go back, and that is through a very special enzyme which I will color code using orange like this. The only way they can go through is via this enzyme, and that enzyme is known as ATP synthase or ATPase for short. This is the enzyme that actually produces the ATP. So tons and tons of hydrogens, tons and tons of protons inside, they can only exit through this ATPase enzyme. So they can exit through this like this. So now we are getting H pluses back in the matrix, and it's these H pluses that go and combine with the oxygen and the electrons to produce water. Okay, now that is complete. We get our final water like this. Let's box that in. Right here, this is what is happening. The only way these H pluses can actually return to the inner mitochondrial matrix, so we can return to the matrix, is via this ATPase enzyme. Now, why exactly do we need these protons to return back to this? Because there's a protonic imbalance. No one likes this imbalance. No one likes imbalances. No. I have spoken too much. I am mixing up my words. Sorry. So no one likes protonic imbalances. No one likes typical imbalances. So that's why H plus has to actually come back down. The only way it can do is through ATPase. Now, funny thing, do you see the structure of ATPase? It's like this. There's this head sort of thing on this side, and there's this huge tube like this. Now the thing with ATPase is this thing can spin really well. This thing is capable of spinning. This little head sort of thing can spin. So when H plus actually comes back down through all of this head spins. And when that head spins, What do you think is produced? When that head spins, we produce the main thing we need to produce, that is ATP from ADP plus inorganic phosphate like this, and this process is known as chemiosmosis. This process is known as chemiosmosis. When the H pluses return, this enzyme spins, its head spins in circles, and that generates the energy to produce ATP. That generates the metabolic reactants, the metabolic pathway to produce ATP. And like we discussed, from this specific stage, we can get basically roughly up to 34 to 36 to 38 ATP molecules. It's not certain. Some textbooks will say lesser numbers, some textbooks will say higher numbers, but it's basically around this range. We are indeed producing a lot of ATP, and that is the final step of the oxidative phosphorylation pathway, and that is the final step of the stage of photosynthesis. The ultimate goal, I'm sorry, did I say photosynthesis? Respiration. So the ultimate goal of respiration is simply going to be to produce ATP, so energy in the form of adenosine triphosphate, which we have successfully done thanks to this ATPase enzyme, protonic imbalances, coenzymes, and all previous reactions thus far, producing a final rough amount of about 34 ATP from all these coenzymes that is coming from one glucose molecule broken down into two pyruvates. That is basically the final end. We will end that with a straight line like so, and that is the final process.